Yeah, nobody cares about that. Everybody pull up the Histology study guide so I don't have to flip back and forth, because most of what I'm going to be doing is read off this and then take a source, one of those three, to find good pictures as I talk. Does that make sense? All right. So make sure you have this up on your phone. And what I'm going to do mostly is going through the things that it says right here that you need to know. This is very different than the lab that I was doing on skin. It had like a long list of terms at the end. This has a long list of objectives. So we need to be able to get through the objectives. What we're going to do is objective, then bounce to a table. Objective, bounce to a table. Insert pictures that help show things. Are you with me? You got the basic idea? All right, so let's start with the easy. Name the 4 primary tissue types in the human body. There are 4 tissue types. They are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Are you with me? I'm actually going to start writing things on the board right now about those tissues. So, if we're talking about epithelial, there are 3 things that we use when we're describing epithelial tissues. And they are, and I said this very briefly in lecture, the layering, the shape of the cells, and any specialization that they have, those cells. And I will tell you right now, in your book, those things are summarized in table 5.1, 5.2, and 5.3, but they're really, really simple to understand. Layering, does it have layers or not? We talked about that before, right? If it's 1 row of cells, we're going to say it is simple. If it has layers, we're going to say it is a stratified tissue. We stole the word stratified from the geologists. They talk about layers of rock as being the rock strata. Good? Yeah? Okay. So that's easy. Then we talk about the shape of cells. There are 3 primary shapes that we deal with. If the cell is tall and thin, I'm like exaggerating over what they often are, and especially if the nucleus is kind of vertical oval because the sides are pressed in on it, we're going to call that columnar because it's kind of like architectural columns. You with me? Okay. Now, if the cell is about the same height and width and it has a round nucleus, we're going to probably call that cuboidal. Because from some angles it'll look cube-shaped. That doesn't mean that it's 6-sided like a block, but from some angles it'll look cube-shaped. You with me? Okay. And then the one right here, well, this is the easy way. Didn't be shocked. I have an empty Mountain Dew can. Oh, come on, you're supposed to laugh because I have Mountain Dew around me all the time. All right, so the S in the word squamous should remind you of squashed. Are you with me? That cell is squashed flat. It's super flat. In fact, it's the only spot where it's bigger, it gets a little bigger so it can fit the nucleus in. Does that make sense? But that is a squamous cell. So those are the 3 main shapes. There are some other shapes, but those are the 3 main ones. Okay? And the third thing is specialization. I already told you that that's the 3rd table in your book. So table 5.3 is on 93. So all that stuff, table 5.1, 5.2, 5.3, those are on pages 92 and 93 in your lab book. And you should know that. Why is that? Oh, because it's pointed up there. Here we go. So this is table 5.2, and this is, those are squamous cells. See how they all look squashed flat? Okay. And these are cuboidal cells. See how they're about the same height? as they are width. I mean, use your imagination, not perfect, but the big round nucleus is a great tip-off. Good? This is a piece of kidney tubule cut lengthwise, I'm sure of it, because that's the best place to find cuboidal. This, do you see tall, thin cells with the nucleus squeezed in between in kind of an oval? Let's trace the outline to make it easier. So we got us a nucleus, and the cell is shaped like that. We got another nucleus and a cell shaped like that. These are columnar, you agree? All right. Let's go one further. These are columnar cells with this fuzzy looking thing here. That's the brush border. Whole bunch of microvilli for surface area. This is intestinal tissue. Does that make sense? Okay. All right. So, but these tables are really nice. They say it really nicely because I'll point out in your study guide, it says to know what the different cell types are, what they're used for, and where they're located. Clear? So we already said that cuboidal cells are in kidney tubules, but I didn't say, and I should have, they're good at secretion and absorption. What are we talking about? Well, most of the filtrate, that's the stuff that'll become the urine eventually, most of the filtrate going through your kidneys, you're going to reabsorb the water out so you don't dehydrate and die. You take that out of the future urine and you put it back into the blood. Does that make sense? Okay. Most of the salt, same thing, you'll take it back in. So you get the idea, different shapes. Now, this one, transitional epithelium, I'm going to mention it because I have it in this table, but then I'm going to go back to that study guide, because this is not one of the 4 epithelial tissues they're harping on that they want you to know. Let me go ahead and write down off the top of my head here. Specializations. Does it have microvilli? If it has microvilli, you know, increased surface area, that would be for absorption. If it has cilia, that would be cause movement. That might be, oh, here's an ovary, and here's the uterus, and here's the fallopian tube, and it's making a current to wash the egg towards the uterus. Does that make sense? It might be, oh, we've got a lining of the trachea with mucus to catch things like bacteria, pollen that we're trying to get down in the lungs and irritate the lungs. Well, we're not going to leave them there in trachea. We have cilia go move the mucus up the throat, well, up to the throat so you can swallow it down the stomach and you can kill this bacteria, mostly. Does that make sense? Yeah? Okay. So, cilia would be for movement. Keratin, the protein, I think they call having a whole bunch of it keratinization. That's it, ization. I'm hoping I'm spelling it right. But that's make it tough. To make this tissue strong and tough and able to survive. Your fingernails are made mostly out of keratin. Does that make sense? Okay. Let's see what other one they had in their 5.3. Oh, goblet cells. Yeah. Okay. So, goblet cells here. These are special. They are weird. They're easy to spot on most microscope slides because they stain really, really pale. See the opening to the outside, the opening to the outside? Those are mucus glands. They are single cell glands. They're one cell that is capable of making mucus. Let's say another way, that's not true. They're capable of making the protein mucin that when it hits water, when it leaves the cell, it becomes mucus. Are we good? Does it make sense? Okay. So those are the specializations they had there. So I will put goblet cell makes mucus. And we use that mucus for different things in different parts of the body, different reasons. In the intestines, you do to lubricate the passage of food. Good. In the In the lungs, I already said it would be to capture bacteria and pollen, keep them from getting to the lungs, trap them before they get there in the trachea. Good, does that make sense? Yeah, so different purposes. All right, but those are the three things they have there. I'm now going, so we did name the primary tissues. That was epithelial, connective, here. Do I have anything blank I can use? Yeah. All right, we're temporarily going to stop annoying me. Here we go. No, we don't. Why do I not? Here we go. This is mostly blank. Now we have space to write on the board. Good? All right, so I talked about epithelial. That was the first of the four major types of tissues. Good? All right. Anybody remember the second one that I said? Yeah, connective tissue. So what I want you to know for connective tissue, we're going to go through some examples. Oh, by the way, how will you practice this stuff? If we don't get through all that, what can help you prepare? Anybody look at what's happening before the end of the week in quizzes? Oh, I'm not going to flip back, just trust me. On the 18th, you have to have the quizzes done for epithelial tissue, connective tissue, and integumentary stuff for this week. Does that make sense? Everybody good? All right, so let's talk about connective tissue, which is the second type of tissue. It has three things. All connective tissue has these three things. Are you with me? Not all in the same amount. And we'll give examples of differences, okay? So, all connective tissue has specialized fibers. All connective tissue has a ground substance. That's the stuff that is in between the fibers, in between the cells. Good? Good? All right. And all connective tissue has specialized cells. Now, those specialized fibers, black, black, black. Okay, that's not working. We've got to get another color. So, let's start with specialized fibers. So I said, oh, there are three types of specialized fiber. Oh, sorry, I said these are the three things: the fibers, the ground substance, the cells. But what I want you to know is that in fibers, specialized fibers, we've got lots of different kinds of specialized fibers. Most of the time, not all the time, there are three of those that we deal with. And I'm going to write those down. That's why it's switching color. Good? So three types. And the ones I'm going to go for are collagen fibers, reticular fibers, and we'll pull up a picture. It makes this more clear. Okay, and then elastin fibers. Collagen fibers, reticular fibers, elastin fibers. But there are others. Good. Some types of connective tissue have other ones. Now, these two are kind of special. The collagen fibers and the reticular fibers are made out of the same protein. I hate the way they named this. They're made out of a protein called collagen. So, collagen protein. But I'm sort of like, why did you do that? If the little fibers have a name that's not the same as the protein, why can't the big fibers have their own name too that's not the name of the protein? But both types of fibers, collagen fibers, reticular fibers, are made up of the collagen protein. Are we good? Elastin fibers, this one makes more sense, are made out of elastin. It's another protein, elastin protein. We only have two spots in the body that would have a bunch of that. Anybody want to make a guess?
Your ear.
Your ear is one, yeah. Here, let's do a quick demonstration here. Hey, I can bend it, snaps back, bend it, snaps back, bend it, snaps back. I can play with my dog's ears all day, right? Okay. You can take it, bend it, bend it, bend it. Look how it snaps back, doesn't matter. That's because we have the elastin protein. It stretches, snaps back. Great. The other place is the epiglottis. The glottis is the name for the opening to your trachea. We have like a little trap door that goes down every time you swallow to make sure things go into the esophagus behind, and then it snaps back when you're not swallowing. So every time you swallow, boing, 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 elastic fibers. Does that make sense? Okay, so that's good. But let's do these together. The specialized fibers and the ground substance together are things outside the cell, around the cell. So we have a fancy schmancy term for them. Together we're going to call those the extracellular, because it's outside the cell, matrix. Good? Those are the extracellular matrix. And some ground substances have one type of consistency, and others have totally different. You would agree this is pretty rubbery, pretty springy. Yeah, okay. The ground substance for bone is a mineral called either osteoid or hydroxyapatite. People use both names. That's tough, like concrete. Do you agree? It's still a ground substance that the fibers are running through. Good, all right. And so you have different. The weird one, and this is one I was thinking of when I was writing this down. Anybody know what the strangest connective tissue is? Like you don't even think of it as a connective tissue, but it is. Blood. Blood is considered to be connective tissue. Now the ground substance is easy. That's the plasma. That's blood plasma. Specialized cells. That's easy. Of course, we got our white blood cells, our red blood cells. We have our fragments of cells that we call platelets. Yeah, we got specialized cells. No big deal. Fibers in blood? But you only see them when you've got a cut that you're trying to repair. Does anybody remember what the word, the suffix on the end of a word in anatomy, ace, means? A-S-E, that we talked about before? If a word ends in ace, it's a? Enzyme. Yes, perfect. Good answer. Name? Gabrielle. Gabrielle's my favorite right now. All right. So yes, if it ends in ace, it's an enzyme. I'm going to give you another one of those. If it ends in ogen, like whatever it is, ogen, that's the inactive form that could generate the active form if you need to. Okay? I'll give you an example. The digestive enzyme pepsin, when it's not in its active form, we call it pepsinogen. Does that make sense? The digestive enzyme trypsin, when it's not in its active form, we call it trypsinogen. Well, floating around in blood, blood has fibrinogen. And as the name tells you, if you activate it, it becomes a fiber. This is a protein that in its normal day, it's floating around in the blood, all coiled up in a little ball shape. Does that make sense? Okay, floating around. But if it gets close to a place that there's a cut, like say this is a cut wall of a capillary, cut wall of an artery, whatever. You got a cut in a blood vessel, and you're going to bleed out there. These damaged cells here, they're releasing chemicals that will activate the fibrinogen, turn into fibrin, and it will go. And then, as the blood tries to wash it out, have you ever seen walking along a path and you see a stream or a river, and there are branches down, and behind all the leaves are piling up, all the trash is piling up, everything. All of a sudden, those fibrinogens, they're fibrin now, and you have all the things like the other blood cells packing into there, getting caught. That's the soft beginning of a clot. Now, some of those things are fragments of cells, pieces of cells that we call platelets. The thing they break off of the big, real full cell is called a megakaryocyte, but you'll learn that in the circulatory. Anyway, in AP2. But when you activate those platelets, they grow all these extensions too. So they start doing the same thing. But the difference is when they're covering the cut with all the little extensions, eventually they're gonna pull them back in. And when they do, that's what squeezes the clot, pushes the fluid out, and starts to make it that denser, hard clot that we end up with. Does that make sense? But the whole thing starts by activating a special fiber. It's not a fiber until you need it, but there is fibrinogen. So yes, blood is still connective tissue, has all three things. Does that make sense? We'll talk more about it later. We'll show more pictures later. I'm going to go ahead and hit the other two. Are you guys okay if I erase this? You know the OGen thing? Yep. Okay. I'm going to give you the two other tissue types. Then I'm going to keep that out of the way so you can take a photo. Then we're going to erase it. We're going to start going for pretty pictures. Good? All right. So the next one, number 3, we've got muscle. And you already know this, but we got 3 types of muscle. In the heart, we have cardiac muscle. And that is involuntary. You cannot control that. All right? We also have, in other places, we have involuntary stuff, like say in the lining of your digestive system, the lining of blood vessels, we have smooth muscle. And that would be, let's call it tubular organs. Like I said, the gastrointestinal tube, the blood vessels, are we good? Yeah? Okay. And those, you don't think about to make it happen, but it does happen. Suppose you're walking around outside and you hear rustling. I heard stories that there was a panther in the area. All of a sudden, the blood going to your eyes, those blood vessels get bigger, so more blood can be delivered. We call that vasodilating. Oh, and guess what? The blood going to your stomach, those blood vessels will vasoconstrict. Because it's more important to run away from the threat than it is to, oh, I'll rest and digest. Does that make sense? Yeah. Okay. So, smooth muscles and tubular organs like blood vessels, like the digestive system. And then the third one, this is the main one, this is one we talk about so much, skeletal muscle. And if you look, I am going to flip over. I know I said I hate flipping over because it takes so long. You're now about to see it. They'll eventually catch up. I promise. Don't make a liar of me. Okay, histology. Here we go. So, what was I going to do? I'm like ADDing. Skeletal muscle. Brain and neutral. Okay, so we're looking at the thing. Here we go. What I wanted you to know, what we're going to do on this study guide, we're going to go over 4 different epithelial tissue types. Those are the main ones I'm going to worry about because those are the ones in the study guide. Those are the ones you're going to see on the quiz because those are the ones in the study guide. Does that make sense? Like, they have transitional epithelium. I might talk about it. I may put it on a full lab test, but I'm probably not because these are the 4 they're really hitting on for epithelium. These are the ones, let's see, 1, 2, 3, 4, 5, 6. These are the 5 that they're really hitting on for connective tissues. There are others, but in muscle they have all 3. So they want to know where they are and what they do. We've already talked about that. I didn't write it all on the board, but you agree we talked about all of it. The little cardiac muscle is in the heart only. It's for pumping blood, yeah? All right, so we've got those, but we'll get those. And then this bottom here, neuron. Oh, I remember what I was doing. I was coming over here because I was going to show you that to help you prepare for quizzes and tests, sorry, the little on-paper quiz and for the test, we have a practice quiz for the skin stuff, for the integumentary, the stuff that Monday and Tuesday mostly focused on. And you and I right now, we're focusing on the tissues. We're doing histology. So you agree we have a quiz here, a practice quiz on connective tissue. We have a practice quiz on epithelial tissue. And we have one on muscle and nerve. But look at the dates. Oh, wow, I didn't. Okay, I thought I made this one different, the muscle and nerve. It's not. I'm going to extend that one because I'm probably not going to put muscle and nerve on the quiz next week. I'm probably going to review the muscle and nerve in more depth whenever we do muscle and nerve, because they have their own chapters. They're not like the others, you know, where we do a quick overview and then get into the stuff. Muscle, we got multiple chapters on muscle, we got multiple chapters on nerve. We can fit that stuff in there, so I'm not too worried about it. Expect this one to change, but these three right here, they're due on the 18th. Get them. Make sure you don't get late fees. How do we not get late fees?
Before the due date.
Use every attempt before the due date. Every attempt. If they're 20, 25, use them all. Then you're not tempted to use them again and get late fees. What do you do if you want more attempts closer to the test to use them for practice?
Let you know.
Yeah, you talk to me and say, I would like more attempts. Can you move the due date for me so I don't get a late fee? And I'll move it and I'll give you another 5 attempts or something. I'll give you more attempts. Does that make sense? That way you don't get hit with late fees. If you just try to do this on your own, like, I'm gonna save 3 and then I'll use them later, you will get late fees. And late fees, it's in the syllabus, they're 5% a day. Well, I don't know if I put that in syllabus, but they're 5% a day. I can show you the 5% a day. No, I can't, because then I'd have to pull up the grade book and show you everybody's grades too. But if I'm in the grade book and I click on the upper right corner where they have like the settings thing, it pops up, I can choose the amount, and late fees are 5% a day. That's for all my classes. That's generous and kind. Last semester they were 10% a day. So, anyway, you get the idea. So the reason I jumped back here was to show you, do not forget that before the end of the week, you need to take the integumentary lab quiz, you need to take the connective tissue and the epithelial one. Are we good? Is that clear? Okay, I'm going to write it up here on the board so it's very clear. Okay. Quizzes. Integ. Oh, there, the dyslexia kicks in. You see it? D's, B's, and G's. I flip them. And P's. Yeah. So that was supposed to be integ. All right. R, connective, and epithelial. These before the 18th. Everybody good? Yeah, so take them, use them, know it. Okay. So, but we've talked about the 3 types of muscle. We talked about where they're located. Yes, we've talked about what they do. I'll say this one again more clearly. I said those are the muscles we usually think about. Those are the ones that when they shorten, they pull on bone and cause us to move, cause us to stand, all the things we normally think of muscle for. Good? All right. And again, you'll get a lot more of that as we go on. So the 4th type of tissue is nervous tissue. And, I'll be more clear, there are 2 different types of cells in nervous tissue. Actually, there are a bunch of types, but we have neurons, I'll call that A. Neurons. And then I have a bunch of support cells for the neurons we call glial cells, which is a horrible name because glial means glue. Back before we knew all the awesome things that they did, we were like, oh, they glue the neurons together, so we call them glial cells. But there are like 6 or 7 different types of glial cells. Are you with me? And when we get to nervous system, you'll have to know them all. Right now, that's not the case. But I will talk about one other thing. Your textbook, all the textbooks, when they talk about neurons, let's see right here. I'm flipping back. We have 3 pictures of neurons. 3 pictures of neurons. All right, so the lab book exists. The lab book has pictures of neurons, and I'm going to flip back to it. Then I'll try to move this. Come on. All right. they have this drawing of a neuron, the classic drawing that's in all the textbooks. I mean, that's a photograph for one, but really does look that way. And here they show them with all the glial cells around them. Yeah, that's great. What I want you to get, and I'll say it right now because I hate the way people do things. In anatomy, they'll be like, this is the neuron, and then you get to the actual chapter on the nervous system, will say, but wait, the thing we told you, that's only part, there's more. So they're going to tell you that this is the shape of a neuron. That's the shape of one of the three main types of neurons. Okay, let's go. So the main shape of a neuron, you have a cell body that's kind of spiky, has pieces coming off they call dendrites. They're like branching things. Usually, not always, those are considered to be the input part of the cell. Then we have a long axon that goes off, and then we have at the end the axon terminals. Okay, great. That's what most drawings show, and that is one of the types of neurons that does exist. But that is a neuron that we would call a multipolar neuron. Multi, because many directions. We've got branches, poles going off every way, and one really big one there. Do you agree? We also have, and none of this matters right now for the upcoming quiz. None of this will matter until the nervous system test, okay? But we also have, coming up, when we get there, cell body, and it's like it's been moved to the middle of that axon. They actually call this whole thing dendrite and that axon to avoid confusing it. But this, would you agree we've got two main poles off that nerve? That's a bipolar neuron. Does that make sense? Not enough nods. Y'all with me? Okay. So multipolar neuron, I'll put out polar neuron, and then we have a bipolar neuron. And then the truly weird one, we have neurons that have a cell body that looks just like a normal cell, and then it has one little extension, and it goes to the axon. You know, like our nucleus is over here, and we have our dendrites here doing our thing, and then we have our axon terminals over here. And this, because there's only one piece coming off of the cell body, we would call this a unipolar neuron. You with me? Now here's the really weird part. Textbooks, until you get closer to the nervous system, act like they're all like this. All your sensory ones are like these two. I think they're pretty important. I would have been mentioning them from the start, but I didn't write the textbook. Are we good so far? Okay. So you know the glial support cells, you got like 6 of them. You know you got 3 different shapes of neurons. You know that neurons and glial cells are the parts of the nervous system. Are we good? Yep. All right, we have done the 4 types, and we've done it in way more detail. We're not just rattling off the names, but objective 1, name the 4 primary tissue types found in the body. We got that. Do you agree? What I'm going to do now, turn this around, try to make that less visible. All right, I'm going to say, everybody take a photo. I'm going to erase. Everybody take a photo. I'll hold it so you can use your phone to take a picture. Okay, yeah, fine. So everybody take a photo real quick, then I can erase all the things on the board. Okay? Y'all good? I didn't see that many phones go up, but we're good. All right, I'm now erasing. Nervous system be gone, muscle system be gone. Connective tissue be gone. All right, we got that. Epithelial. So we've done good work so far. We've covered a lot of details. Let's now hit the next objective. The next objective, note the function location of the 4 epithelial tissues listed in Table 1. So I'm going to scroll down to Table 1, and it wants you to know about simple squamous, simple cuboidal, pseudostratified columnar, and stratified squamous. There are other epithelial tissues, but those are the ones in Table 1. Yes, you guys see that? Okay. We're going to talk about them, but before we do, we're just going to do a real quick review. All right, I'm going to draw a tissue, and you tell me what it is. We got a basement membrane here below the tissue. These are our cells. What kind of cells would these be? Oh, yeah, but the full name of the tissue would be simple cuboidal, because it's one layer. Does that make sense? So, yeah, cuboidal would have been a half credit answer. Simple cuboidal would have been the full credit answer. Good? All right, now let's modify. What kind of tissue do we have? Remember, tissue is both the layering and the shape of the cells. Louder? Yeah, you got simple columnar, exactly. All right, now, what do we have? Stratified columnar. Everybody good with this so far? Now we're going to go for the weird one. This is not one of those three simple shapes that we've been talking about. This is one that messes a lot of people up if they're not paying attention. So, I'm going to draw tissue. It's one that a lot of people think has layers and it doesn't. Cells have weird shapes. We'll do a few of them here the easy way, then we'll make some that are a little more realistic and strange shapes. Imagine you have a mixture of cells. It might be like, think of them as wine goblets and upside down wine goblets for the basic shape, like that. Then you got a couple weird things that they don't even go to the top, not even up to the apex. We've got this going on. Anytime you have a cell with a big, fat, thick area, that's probably where the nucleus is going to hang. All right. Now, you look at this. Suppose you were lousy with a microscope. You didn't know how to focus. You couldn't get the membranes visible very well. You'd see row of nuclei, row of nuclei. You said, that's stratified. It's not. How do you know it's not? They're all attached to the same basement membrane, all attached to that same collagen fiber at the bottom of the tissue, yes? Even ones that don't go all the way up to the apex. These are all one tissue because they're all attached to one layer at the bottom. Good? So you know it's simple. But because it looks stratified, we have a special name for it. We're going to call it pseudostratified. In fact, because they're tall, you could like maybe go pseudostratified columnar. In fact, let's change one of these because it looks so much like it. Let's say this is light staining. I probably should have used black. That has a bunch of granules in it. This would be one of those single-celled glands. Give me a G name. Goblet cells. Perfect. And they make what? Give me an M. Mucus. Yeah, they make mucus. If you had this, let's add one more detail. We're getting a specialization. And I know with my drawing, it's hard to tell the difference. So I'm going to draw the feature, and then you tell me, I'll tell you it's one of the two, and you tell me which it is. So we got this thing going on the top. You think we're dealing with microvilli or cilia? Well, actually, this one's supposed to be cilia, but my drawings are rubbish, so that's fine. But this, goblet cell laying out mucus, all those pseudostratified cells with the cilia, that's the lining of your trachea. Does that make sense? Oh, so let's say it another way. Hey! Pseudostratified columnar, item 3 on table 5.1 at the back of your study guide. You need to know that. What is the function? It moves the mucus with all the nasty things trapped so it doesn't get into your lungs. What is the location? Lining of the trachea. Everybody good? All right, good. So we've got that. We've done one of those. Let's now do the others. All right. Let's try using your book, your lab book. If not, we'll very quickly move to the lecture book where everything looks better.
I'm sorry, what was the function of the simple cuboidal?
I was just about to do that one.
It was the
We haven't done simple cuboidal. Oh, sorry, simple cuboidal we did. It had, I thought you, for when I was thinking simple squamous. Simple cuboidal is absorption and secretion. And it said that in your lab book on,
The one that you just drew, wasn't that?
page 95. That was pseudostratified columnar.
What was the function of that one?
It's protected because it moves the mucus. Okay. Here, I'll take your lab book and put it right there. Pseudostratified columnar epithelium, protection, secretion, and movement of particles when the epithelium is ciliated, as it is with all the cilia sticking up in the lining of your trachea. Good? Right. So location, trachea, function, protection, secretion, movement of particles. Are we good?
Yeah, where was the location of the simple cuboidal?
Oh, okay, thank you. People are pulling me forward and back. I was trying to say that, but I pointed out before, I said, oh, this is a nice picture right here. This is simple cuboidal. I said, that is a tubule cut longwise. Anybody remember me saying that? Anybody? Nobody. I promise I said it because I'm going to then show you simple cuboidal when you cut across the tube. The wall of the tube is made out of simple cuboidal. That's a kidney tubule. Good? Good. All right. And if you look, simple cuboidal is one of those things you can mess up very easily because it's not, more than any of the other tissues, this is a use your imagination when we tell you it's cuboidal kind of tissue. Okay? Let me explain. There are some that very much, I'm tracing an outline here, resemble a cube. And they have a nice big round nucleus. But there are some that's very much imagination to call that a cube. So what do we know? If it's kind of the same height and width and the nucleus is round, we're going to call it cuboidal. Even if it clearly is a trapezoid shape, because we don't have trapezoidal tissue. Does that make sense? That's still simple cuboidal, even though it's not looking very cube-shaped. This one's even worse. Look at that. Y'all with me? Yep. But that's simple cuboidal. And this, again, is still kidney tubule. See, it says kidney tubule. The other one, I don't know if it said or not, but it's still kidney tubule. Good? Yep. So a location for simple cuboidal would be kidney tubules. There are other places, but anywhere you need to do a lot of secretion and absorption. It's good at absorbing things. In fact, when we drew it on a PowerPoint, we had it looking like the lengthwise cut, but the thing that helps it to be good at absorption a lot of times, of course my marker fails at the moment, it has a lot of microvilli to increase the surface area at the apical surface, so it can absorb things. Does that make sense? Are we good? Am I getting nods? Are you with me? All right, good, good, good. But simple cuboidal, good at secretion and absorption. All right, now simple squamous. Simple means it has how many layers? One, exactly. So let's see if they have a good picture here. Yeah, okay, so your lab book has rubbish pictures. We're going to go for your textbook instead. No, these are actually pretty good. All right. So, here we go. Yeah, this is a good picture. Right there. I'll move the camera instead of moving the book and having the book fall off. This is lung. This is the air sacs in the lungs. Let's talk about that. Here, you have capillary. And here you have air. Look at how thin the barrier between air and blood is. It's 2 cells thick. One flat, simple squamous cell makes the wall of the alveoli, the air sacs, and another one makes the wall of the capillary. So you have 2 right there. if you're only two very tiny flat cells wide, it's easy for oxygen to go from the air to the blood, and for blood to send the carbon dioxide back to the air, so we can breathe it out. Does that make sense, y'all? Okay. So where do we have simple squamous? Anywhere we have to do filtration and absorption, but it's really good at filtration. Air in the lungs, but also the glomerulus, the filtration membrane of the kidney. I'm gonna read the way they describe it, and then we'll move on. So location, air sacs of the lungs. Oh, I forgot to say it, lining of the blood vessels. Well, I did say it. The capillary wall is only one cell thick. Remember I said that? Here's the deal though. You'll get this when you get to circulatory, when you get to AP2. Most blood vessels have 3 layers, okay? The inner lining, which is super slippery smooth, it's simple squamous. And we used to call that the endothelium or the endometrium, good? Then, in the middle, we have the myometrium. That is the thick layer that has the muscles that do the vasoconstriction, vasodilation, yeah? And then outside that, we have the tunica externa, which is the outside sticker. Okay, full connective tissue, pulls things together. Capillaries are so thin to be good at gas exchange, they have lost the other 2 layers. They didn't have them. They're just that smooth inner lining. Does that make sense? And that lining, it's not just the blood vessels, it's also the lymph vessels. The serous membranes that we talked about before, the one around the heart, remember the pericardium, and the one around the lungs, the pleura, those are made out of serous membranes. So they're very thin and they just have fluid they trap for lubrication. Does this make sense? Are we good? All right. So that's your location. And in case you can't see it very well back there, I can change the contrast. But there's the nucleus. It's very squashed flat. Just like in the columnar, the nucleus is squished vertically. Does that make sense? Everybody good? All right. So this is simple squamous. Tell me how good you think your lungs are. What would happen if you inhaled a bug and it got all the way down your lungs and it's scratching the lining of the lungs, the alveoli? Do you think they'd survive that very well? No, because it's very, very weak. It's only 1 layer thick. You rip that layer, and now you're a capillary in blood, and oh, big mess. What do you use if you want lots of protection? You want lots of layers. Am I right? Okay. So stratified squamous is the one we use for lots of, and that's number 4 on that table, the table 5.1 that we're talking about. Stratified squamous, this right here is stratified squamous, the lining on the inside of my cheek. Why? Because if you're chewing on food and it's scraping the inside of your mouth, if it was simple, you'd start bleeding as you scrape it. Does that make sense? So you're eating potato chips and you get a little scrape. Ah! Well, we don't want that. So we have layer after layer after layer after layer. Good. This is also stratified squamous. It's just that this is keratinized stratified squamous, so it's tougher than the non-keratinized inside your cheek. Are we good? Does it make sense? But anywhere in your body you need abrasion protection, you're going to go with stratified squamous because, like the skin, okay? Skin, the outer layer, the stratum corneum, it can have like 20 cells thick. So if I scrape 1 or 2 of them, if I run into a desk or something, man, who cares? Because I've got a layer under that, and under that, and under that, and under that, and under that. And that's just the outer part of the skin, the stratum corneum, the outer part of the epidermis. Does that make sense? Okay. The next chapter, they talk about how the epidermis is the stratum basale, and then the stratum spinosum, and then stratum granulosum, and then stratum lucidium, if you have it, that's only a thick skin thing, and then the stratum corneum. And the corneum by itself is 20 cells thick usually. So that's the power of stratified. You scrape some of those off, it doesn't matter. You got so much underneath to protect. Does that make sense? So that protects your cheek, but let's get gross and go the other end. Say you have to go to the bathroom and you're passing poop, passing feces out of your body. What do you think the rectum is like? It's going to be stratified squamous, right? Right? Because abrasion or resistance. In fact, there it's more important. See all the nasty bacteria with it too. Yeah. Yes.
So is the stratified squamous, is that the most abundant and?
That's a really good question. I don't know. It's certainly the most abundant in places you have abrasion. So the lining of the the lining of the rectum, the lining of the mouth, lining of the esophagus, the outside skin on your body, the lining of the female reproductive tract. Of course, you got a lot of it there. Overall, I don't know, because you really don't have that in places you don't have rubbing. So, say between the gastrointestinal tract and the body wall here, I don't think you'd have much of that. Does that make sense?
Yeah, it's just because I figured that everything, like it's your body, you need protection.
Well, there are some things we do, but we just talked about how
Right.
the serous membranes are simple instead of stratified. And remember, so like around the intestines, around the liver, that was peritoneum, that's serous membrane, that's gonna be simple squamous.
Okay.
So, but I will say there's a heck of a lot of it. It was a good
Right.
hypothesis. Does that make sense? Everybody good? Yeah. All right, so would you agree we've talked about stratified and where it is? We've talked about pseudostratified and where it is. We've talked about simple cuboidal and where it is, yes? We've talked about simple squamous and where it is. Hey, I think we've taken care of the epithelial stuff. Let's go back and look at that objective and see. Yes, there are other epithelial tissues, but those are the ones that are on that study guide. Now, if there are more on the practice quiz, I might put the things in the practice quiz on the paper quiz that'll be next week, or on the test. Basically, I wrote those quizzes, the practice quizzes, to help prepare you for what I'm going to be doing in tests. Does that make sense? Anyway, so know the functional location of the 4 epithelial tissues listed in table 1. I think we did that. Good? Next objective. Know the functional location of these 6 connective tissues listed in table 2. So I'm going to jump down to look at the 6 connective tissues. And the ones that has are areolar, adipose, reticular, hyaline, osseous, that means bone, and blood. So let's talk about them. When you see areolar, think open to the air. I know it's not spelled the same as AIR, but what I'm getting at is that areolar tissues have lots of space in them, okay? We're in connective tissue, so before we do that, I'm going to talk more about connective tissues. I said I would tell you more about it. What were the 3 types of things in all connective tissues? Specialized what? Fibers? Yep, perfect. Specialized cells, yes? Well, all of the connective tissues have all of those 3 things, but not in the same amount. Some of them, like bone, lots of ground substance. That's what it's famous for. But it has fibers too. You just don't usually see them. Some of them, and I'm going to go for pictures right now to show you some of them, we'll move on. Okay? So, if we're looking here, that's mesenchyme. We don't care about that. We're not testing on that. Okay, so connective tissue proper. Here we're going to have areolar tissue. This is in your textbook because there are better pictures than your lab. This is in your textbook on page 169. I would recommend writing that down in your notes so it's easy to get to. But this is areolar connective tissue, and it looks very open, like there's nothing there. Like you've got fibers for sure, and you've got specialized cells living there. This is not nothing. This is not air. This is ground substance. There are things there. Does that make sense? But it's got a lot of space. And when you look at this areolar connective tissue, it has elastic fibers, it has the bigger, thicker collagen fibers. See how here we have the elastic fibers are thin, little stretchy things, and then the thicker ones, those are the collagen fibers. Everybody good with that? Okay. This is areolar connective tissue, and let's just do what they say right here, talking about it. Structure. Scattered fibroblast. Oh, we're going to introduce a new suffix. Everybody ready for this one? When you see blast, I'm going to give you two words that I want you to think of. Okay? When you see something glass, the B should remind you of baby, because that cell might grow up to be something else. Okay? But it should also remind you of builder. Because there are no child labor laws in anatomy. We put the baby to work. Does that make sense? So a fibroblast, you probably guessed, builds fibers before it grows up into something else. Does that make sense? Okay. Guess what? In bone we have an osteoblast. What do you think it does? It builds bone matrix. Good. In cartilage we have a chondroblast. What do you think it does? It builds cartilage. Are you with me? Is it making sense? Okay. But blasts are baby ones. And you can see we've got, both in the drawing and the picture, we got a whole bunch of those specialized cells, right? And what are they going to be doing? They're going to be making substances where it says here that the areolar connector shows abundant viscous ground substance. Think of it as being like a jelly, okay? When we talk about viscosity, we're usually talking about things like how thick is the oil in your car. I know I'm not supposed to talk about cars. Well, if it's really thick fluid, like a jelly, does that make sense? So it's got some give to it. It's not like solid, like bone. So ground substance doesn't have to be solid. But you already knew that because what's the most fluidy of the connective tissues? Blood. Yeah? So blood plasma. Anyway, but it's viscous. It has a few elastic fibers and collagen fibers, a lot of blood vessels. Okay, great. This is what it looks like. Protects tissues and organs, binds skin and epithelia to deeper tissue, space for blood vessels and nerves. Since there's a lot of blood vessels in there, anywhere we need to pass blood vessels from one area of the body to another, we might have some areolar connective tissue around it, okay? Protects tissues and organs? Yeah, well, if you got all that gooey, squishy kind of ground substance, if you surround things that you don't want to have smacked into, there's a little give, a little cushion. Does this make sense? All right, so location, the papillary layer of the dermis. All right, now, this is something that is in the next, and we're going to use the good book. I know we'll use your lab book, because this picture is pretty good. Remember, this is the thing I was talking about with the other class earlier this week. So let me jump to skin. Here we go. Oh, heck! Okay. We could use this picture, but we can get bigger because we have the same models. We're going to use the model, okay? So I'll just say this really fast. They said the papillary layer. Okay, so when we are looking at the skin, everything on this purple and above, this is epidermis. Everything below it, down to the yellow here, this is dermis, this is epidermis, this is hypodermis, below the dermis. Are we good so far? Okay. So, when we look at that, I already told you that the top, the epidermis, has layers that we call the stratum corneum. That's the one on the outside that's in pink. And below that, the ones in purple will be the stratum, well, this, because it's thick skin, has the lucidium. Lucidium is only found in thick skin. The stratum corneum, lucidium, and then here we have granulosum, spinosum, and at the very bottom, at the base of the epithelium, we have the stratum basale. So those are the layers. And the mnemonic to remember is come, let's get sunburned. In order from the outside where the sun is to the inside. Does that make sense? Come, let's get sunburned. It's stratum corneum, lucidium, basale, spinosum. That's why I said that. Granulosum, spinosum, basale. Good? Yes? All right, good. So there you got that. But here, do you see, let me a little pointer. Here we got a little sticky uppy part of the dermis, a little sticky uppy part of the dermis, a little sticky uppy part of the dermis, a little sticky uppy part of the dermis. Those things that are sticking up, the anatomists who saw them, they thought they looked like fingers sticking up, so they called them dermal papilla. And the top part of the dermis is the papillary layer. Good, yep. Anything below that is full of reticular fibers, and that's the reticular layer. Are we good? And that's like 80% of the skin. Let's pull up your lab book again and show the picture. All right, so you can see the papillary layer is this little tiny part right up here where the dermal papilla are, good? But most of the, okay, that's not making me super happy, I'm gonna go for the other book. But most of that is the reticular layer down here. Let's go for the other book that has a prettier picture, as it usually does. All right, so here, yeah, this is a much prettier picture. Check this out. So the papillary layer is that little part at the top of the dermis where the dermal papilla is, yes? And then most of the dermis is the reticular layer. Are we clear? All right, now let's go back to what we were just looking at in histology and what they said. Right there. Where is it areolar connective tissue? In the papillary layer of the dermis, the subcutaneous layer below the deep to the skin, we're talking about down that hypodermis area, okay? Around organs, nerve cells, some muscle cells, we have some of this around them just to cushion them, not have things harass and get to them. Does that make sense? All right, so we've got the first of the ones on there, areolar connective tissue. The next one is the adipose tissue, and that's the next picture. This is a perfect picture because it's a drawing. This is a photograph, so it's more accurate, but it's hard to understand what you're looking at. Let me make this a little darker so it's a little easier. When you look at all the white, no, do not do that to me. When you look at all the white things here, you might be thinking to yourself, oh, well we said all the white stuff here was the ground substance. That must be the ground substance. No! There's almost no ground substance. Remember, I said that some tissues have more of one, more of the other. Adipose tissue is almost all cells. These cells, I'm going to make it lighter now so you can see the picture again. Look at these specialized cells. All of the nucleus and cytoplasm is shoved off to the side. There's a big lipid storage vacuole here. That is lipid storage. The reason it looks white, do oil and water mix? No? Okay. Well, guess what? Most of the stains we use in anatomy are water-based. So, since they're dissolved in water, if you cut a piece of tissue and you put it down a microscope slide and some of it's fatty, and then you put the stain on it, you rinse off, none of it stuck. None of the stain stuck because it was dissolved in water, and water doesn't like fat. So it looks white on the slide. It's not that there's nothing there, it's full of fat. Does that make sense? Okay? So, but what I'm getting at is these are all three connective tissues. And you can see when you're looking at the areolar, a lot of space and a lot of fibers. Are you with me? You have cells, but they're not the main thing. Now you look here, you look at the adipose tissue, and you've got all three things, because you always do. But it's mostly cells. There is space, but there's not much ground substance, and you'll be there's more there, and you'll be really hard pressed to find the fibers. You have to look a long time on a slide to come across a fiber, because there's not much room. Does that make sense? Everybody see it? Okay. Now this is reticular fiber. I told you that there were three types of fibers. You may remember the three types of fibers. I said three main types of fibers. Collagen, reticular, and elastin, yes? Okay? So this has a lot of reticular fibers. Those are made out of collagen, but they're really fine, diffuse, spread out. They're the scaffolding that these white blood cells are crawling around on, hanging out on. This picture tells us this is the spleen. But it could have been a lymph node too, part of a lymph node. But so you have these reticular fibers, you have this space in between, you have these white blood cells hanging out here. You know what's going on there? It's part of the lymphatic system. You've got all the lymph going through all that open space, and the white blood cells are sitting there just watching. I know how to explain it. This is an old movie. It used to be like, I was using this because this is a new movie, but I'm so old now that it's an old movie most of you guys have never seen. Probably came out before most of y'all were born. Anybody ever seen Will Smith in Independence Day? Okay. All right. Well, here's the short version. He flies a UFO that was at Area 51 up to fight the aliens in space. The end. Okay? That's the whole plot, whatever. And there's a spot where he has to fly the ship they stole from the aliens up into the alien mothership. You with me? That alien mothership is full of steel girders in every direction with little fighter UFOs hanging off of them, watching to see if something suspicious goes by. And of course, once they spot him, they all come off of it and chase him through the mothership, you know. So, but that idea, all these white blood cells are hanging out on the reticular fibers, looking for bacteria and bad things coming through.